I'm going to be talking about these systems neuroscience approaches to understand motor learning and recovery post-stroke. So in my lab, we are essentially a translational neuroengineering lab, and we are looking at ways in which we can remedy the disability or at least somehow improve disability in stroke patients. I do know that you've had a talk on stroke earlier this week by another speaker, but I still wanted to take a minute to introduce the problem here. So these are US statistics, but stroke is it approximately occurs, a new stroke occurs every 4 minutes in the United States, and it's very, very common all over the world right now. There is a lot of cost associated with this. The cost associated with the disability. A lot of patients, they go on to have permanent disability after a stroke, especially of the hand and the finger movement, the dexterous movement. So it's a huge problem worldwide. And what we are seeing of late is that people get to the emergency within time. Earlier, what we used to see was that there was a lot of casualty associated with stroke, but now people go on to live, but they don't go on to have good fully functional lives. They have a lot of disability because we've had some improvements in getting them care. And one more thing about stroke is that stroke could be ischemic, wherein there is a clot that is formed in one of the arteries of the brain, disrupting the blood flow to the different brain cells or in different areas. Or it could also be hemorrhagic, where some vessel in the brain ruptures, which also leads to similar consequences. So this is a sort of a schematic, a cartoon. Typically in your brain, your motor actions are instantiated by a lot of different areas. For example, there is motor cortex, there is thalamus, basal ganglia, cerebellum. All these regions are involved in motor control and motor learning. And after a stroke, there is a lot of network activity that changes in this region. And just the way the blood vessels in the human brain are designed, most of the strokes end up occurring in this middle cerebral artery, and there is a lot of motor disability as a consequence of this. A majority of patients go on to have motor disability. So I'm gonna be showing some of the experiments that we do with this particular stroke in mind. So we have an experimental animal stroke injury model, and we go and experimentally lesion the motor cortical area of the rodents. So what happens after a motor cortical stroke typically is that there is trouble translating intention into action. If you've ever interacted with one of these patients, they do have the intention to move, but they cannot translate this intention into action. The typical sequelae of stroke, the typical sequence of events that happens, that a clinician would describe is that there is a lot of weakness in the limb, there is spasticity, there are slow movements, the movements are variable, there's a dystonia, which is abnormal muscle co-contraction. And there are also reports that there is poor learning in the system. Stroke patients cannot learn new skills like healthy individuals would. And as a lab, we are looking at what is the circuit basis of this. So as a systems neuroscience lab, we are investigating some of these circuits using electrophysiology tools as to what is really going on in the circuits in the subcortical areas or in the perilesional cortical areas after a stroke. So my lab is looking at this problem through 3 lenses, if you will. We essentially work with rodents as well as human patients, and we are looking at 3 scenarios. So in terms of motor learning, we are focused on neuroprosthetic learning or brain-machine interface learning, in which the subjects learn to control artificial or external disembodied device. In this picture, it's a robotic arm, but this could also be a computer cursor if somebody has speech impairment. Then we also look at motor skill learning with the overt limb, where you're actually using your limb. So in this case, I'm showing here, this is a reach to grasp task in which the rodents actually learn to use their limb, either the left or the right limb, based on the handedness, to pick up a sugar pellet. And then we are doing both these tasks in rodent and human subjects when they're trying to relearn after a stroke-like injury. And I also want to highlight here that we are interested in both online processes as well as offline processes. So by online, I mean awake practice related things that are happening in the brain, and by sleep, offline, I mean the sleep processes. So sleep has long been known to play a role in learning and memory. And some of our new work, ours as well as others' work, is now showing that that there is a lot of uh, that there is a lot of memory related things with regards to these motor tasks that is also happening in sleep. And obviously at the core of all these tasks, we are evaluating neural plasticity through electrophysiology tools. So with that kind of introduction to what the generic outlook of my lab, I've organized this talk into three parts. There is a brief neurophysiology background, and then I want to focus on one story, which is on the reach to grasp task and the awake processes that we see in the brain. And within that, I'll be showing you what we see in the brain in healthy rats, what really happens during motor recovery when the rats are injured and they're trying to relearn this reaching task. What kind of concordance do we see in the human subjects? And then obviously the translational neural engineering part, how we can modulate this activity then to recover function after the injury. So I'll start with the neurophysiology background. So this figure essentially encompasses all approaches in electrophysiology. There are varying resolutions. I want to highlight two things here, the local field potential and single unit action potential. So we are currently capable of recording these things through intracranial electrodes. So this is shown here within the human brain or the rodent brain. We can put these intracranial electrodes. These are penetrative arrays. And then we can read the single unit action potentials, which I'm also going to call spikes. And then local field potential is also recorded at the same site. All we need to do is just filter the signal in a different range and we can get the more broadband signal. And the local field potential represents the population activity in the region. Another thing I want to point out here is ECOG, the electrocorticography, because this will come up in the talk later. So electrocorticography is shown in this picture here. So electrocorticography is, this is an easier technology to implement in humans. So here you can see over a human brain, this electrocorticography grid. And these, in this case, these are not intracranial, but the recording sites are sitting on the surface of the brain. And these are typically used in epilepsy monitoring units. So the patients who are at risk of getting a seizure, they are monitored via ECOG grids, and I'll be showing you the concordance of our results in rodents in human subjects in which ECOG recordings were done. So I also wanted to bring this slide here to show that there have been studies that have already been done for stroke recovery. I know earlier in the week you had a talk on TMS related approaches to rehabilitation. In terms of recording technologies, there are these different recording technologies, which was also described in the previous figure, but here it's kind of divided into the varying spatial or temporal resolution that they offer. And within the circle are the technologies that I'll be talking about. So the single unit recordings obviously have a great spatial resolution if you compare it to, say, EEG or a non-invasive technology. It also has good temporal resolution, but fMRI and all, which fMRI and techniques such as near-infrared spectroscopy don't have. And some of these tools have been used for assessing stroke recovery. For example, in this case, this group in Kansas, they've looked at non-human primates. So they've looked at this brain maps, the motor maps in the monkeys. And what they see is that post-stroke, the representation of some of the digits really reduces. And this has also been looked by different groups in fMRI. And here also you can see that compared to healthy subjects, stroke patients immediately after stroke have less activation of this parietal areas, but it increases late, few days after stroke, but it still doesn't match up the unaffected arm or the healthy controls. And similarly, people have also looked at the process of recovery in EEG. And here you can see that sorry, I did not put the key here, but post-stroke, some of the EEG oscillations also suffer. So people have studied features of recovery amongst all these different modalities. And I'll be focusing on slow oscillation. So I wanted to put in this slide here. It's a bit of an oddball, but just bear with me here. So these low frequency oscillations in electrophysiology, people have described them in other motor tasks also. So in this study, during monkey reaching, when a lot of muscle activity is there, you see this low frequency potentials, these low frequency oscillation, less than 4 hertz. That are visible in the LFP, and it seems to also bind the motor cortical neurons. And over here, you can see that the same LFP has this low frequency oscillation every time a movement occurs, which is indicated by this downward arrow. And it tends to correlate with this bell-shaped velocity profile. So this low frequency oscillatory activity has been described in non-human primates, and it seems to be important for organizing the movement at the level of kinematics as well as EMGs. So hopefully that was enough of a neurophysiology background. So I'll next delve into what we see, what kind of oscillatory activity are we seeing in our rodents when they're learning a skilled reaching task. So this is the task we spend a lot of time looking at. So in this task, rodents make a movement and they grasp a sugar pellet, which is a few millimeters away. And it has 3 very specific phases. So I'm going to play this video for you. So there is an extension of the hand, then there is a grasping of the sugar pellet, and then there is a retraction. So it's also called reach to grasp task. And we see that typically within 5 days, they learn this task. So this is a healthy rat that is very proficient at the task, and it's reaching for the sugar pellet. So I think I need to disable my laser pointer to play the video. So yeah, so this is on, this is once the rat has become very proficient in this task, and it didn't take the rat a lot of time to reach for the pellet. Typically, once the skill is consolidated, it only takes them around 200 milliseconds to complete this action. So what do we see in physiology? So once the rats become proficient at this task, so we've implanted them in this motor cortex, and when we look at the single unit action potential, so here you can see 171 neurons from 4 different animals, which are lined in relation to the time to reach onset. And you see this nice tiling of activity in the motor cortical neurons. A lot of neurons are active around the grasping. And along with this, what we see is that the local field potentials develop this low frequency quasi-oscillatory activity. So I'm calling it quasi-oscillatory because there's only 1 or 2 cycles. So it's not an ongoing rhythm, but right around the time of reach, there is this LFO oscillation, and it seems to bind all the neurons. So there are 55 neurons in this one reaching trial. And you see that the different neurons are active around the time of reaching or grasping or retracting. And this activity is modulated by this LFO, the quasi-oscillatory low frequency oscillatory activity. And I want to mention here that even though this is one trial, we really see this activity emerge later in learning on day 5. This is not present on a time matched trial on day 1. And over here, you can see, so this is another way of looking at it, but this is the power spectrum from this LFP, all the LFP channels from all the animals. And we see that there is an increase in low frequency oscillatory activity from 1.5 to 4 hertz. And how this LFP is modulating the spiking activity, there are several ways of looking at this, but the analysis we commonly employed to study this is called spike field coherence. So it's a multi-taper method. It's a frequency domain analysis. And here again, we see that the neurons are really getting modulated in this low frequency range by this LFP. And that the parameter, the quantification of this is called coherence magnitude. And we see that this coherence magnitude is really high once the rats become proficient in this task. So it looks like this LFO activity can serve as a marker of learning in skilled, can serve as a marker of skill consolidation in healthy rats. So this is just a cartoon view of this. So as the rats are transitioning from this unskilled to the skilled level, we see that the spiking activity becomes very, very organized, which is indicated by these black rosters. The field potential also becomes very, very oscillatory in this low frequency range. And the movements become very, very fast, consistent, and stereotypic. So what really happens after stroke? Is something, does something really change in this oscillation once a stroke occurs? So for this, we essentially have two stroke models in the lab. But what happens with these stroke is that you immediately impair the performance. So once these rats have become proficient in the task, we go and do the stroke. In this particular slide, the stroke model is the photothrombotic stroke. So in the photothrombotic stroke, we inject rose Bengal dye into the bloodstream of the rat through femoral vein cannulation. And this is a photoactive dye. And then if we shine light of a particular wavelength over any region of the brain, we can cause micro-occlusion in the vessels there. So we shine the light over the digits area of the hand, and we see deficits. But slowly there is recovery. The animals do tend to recover, but there is huge variability. Some of the animals have a more chronic deficit in the sense that they don't end up recovering on this task. So let me also play a video here of a stroke rat. So as you can see that there are multiple attempts to reach and grasp the pellet, but the extent of reaching hasn't really come back. But during the course of recovery, what we see is that the reaching ability starts to come back, but with a motor cortical stroke, the grasping still continues to suffer. So even though they can finally start making contact with the pellet, the grasping motion, that seems to be really, really driven by M1, and that takes the longest to recover. And sometimes it does not recover. In some of the rats, it does not recover. So I'll go to this next slide here. So we do both, we do two kind of stroke models, the photothrombotic stroke. Photothrombotic is an experimental model. Like I described, we shine the light after injecting this photoactive dye. But in addition, we also do this middle cerebral artery occlusion model in which we go and tie up the artery, the middle cerebral artery. And the advantages of this model is that this is more human mimicking. This is what happens in the humans. But this model is not that amenable to physiology because this essentially wipes out the whole lateral hemisphere in the rat. And sometimes we don't get a good signal in whatever is left in the cortex. So we don't get to record from a lot of neurons, but at the same time, we can go and validate our findings in a more human mimicking stroke model. With the photothrombotic stroke, the advantages are it's a more experimentally tractable model. We can shine the light on a particular area of the brain, and we can have very well circumscribed controlled lesions. So it's a more experimentally tractable model. So here, I'll first describe some of the results with the MCS stroke model in which the deficits are mostly permanent because it's a supermassive stroke. So here is this one rat in which MCS stroke was done. So let me unpack this plot for you because there are a few things on this. So here are 71 trials before a stroke in this rat. And you see that as the rat becomes good at this task, there is a lot of 1.5 to 4 hertz power, the LFO power that I just talked about. And over here in the green line is the accuracy at grasping. So this guy is really cruising along and there's a good 60–70% accuracy in grasping the pellet. And immediately after stroke, we see that the accuracy drops. They can no longer pick up the sugar pellet. And concomitant with that, this oscillation goes away. So this is our first hint that this oscillation might be, this could be a marker for skill learning. And once the skill is lost, you lose this oscillation. And this is seen across 4 rats. The success drops, and we also see this power reduction. So here is the pre-stroke power in these 4 rats, and post-stroke, this 1.5 to 4 hertz power really, really goes down. And this is again an example trial from one animal. We don't see that nice LFO, low frequency modulation in the LFPs or its modulation of the neurons. The nice tiling-like activity that we were seeing in healthy rats, we see that barely Few neurons are active around reaching. So these are 53 neurons across 4 rats. And the spike field coherence also at a population level, it reduces. So all the parameters that I was showing you previously, they all suffer right after a stroke. So what happens during the course of recovery? With the photothrombotic stroke, we tend to have recovery, but you can see that there is a huge variability. Um, you see that some of the animals have a poor recovery, whereas some of them have a more quicker recovery. So with this model, we can really see like what is happening during the course of recovery. Does this oscillation really get rescued? And the quick answer is yes, it does. So here is a stroke rat, and you see early after stroke. So this is session 1 or closer to the, starting from the first trial. And this is session 12. And you see like slowly, so these have occurred across a few weeks. And you see that slowly as the performance starts to recover on this task, this oscillation seems to track recovery. So this LFO oscillation can be, it tracks recovery, and it can be a biomarker for recovery. And we see this at a population level as well on the LFP power. Early after stroke, this power is diminished. Later, this power starts to come back when there is a lot of recovery in the rats. And we are putting these 32 channel arrays in the perilesional cortex. So what you're seeing here is the average of what's happening on all those sites. But if we look at the grid, so here are 16 contiguous grids. And you see that this is not just a result of select few channels, but the power really starts to come back in this region, and it's spread out spatially in this region, and it's a consequence of coming back on almost every channel. And this is the same power plot, which shows that there is an increase early after stroke to late after stroke in this particular power. And this is a regression which shows that this power change is really correlated to the success change on a day-to-day basis. So as this oscillation is coming back, the capacity to start grasping again is also coming back in these rats. And this is, again, some of the single unit activity. Again, these are the example trials. You see that the neurons are not really firing. There is no LFO. And later on, this is another example trial that the LFOs are coming back and the neurons have started firing again. This is, again, the tiling plot that I've been showing you before. And you see that later on, more and more neurons start to become active around reaching again. And the spike field coherence also rescues. So early after stroke, the spike field coherence is low, but later after stroke, the spike field coherence is high again. So, so once we established this, we wanted to check the concordance of our results in human patients. So this work was done at UC San Francisco. So we also had access to patients at the medical center, at the hospital. Like I was describing, like patients at risk of seizures are monitored in epilepsy monitoring units via the ECOG grids. So it's very rare that a stroke patient will be implanted with this, but we were able to assess a stroke patient who had this ECOG grid. So this is the patient's MRI and diffusion tensor imaging, which is showing that this patient had a supermassive stroke. This was a very sad case. Typically, stroke occurs in old age, but this was a 35-year-old patient. And this recording was done 4 years after his injury. So that's also something to keep in mind, that there has been enough window for allowing any, so this is not in the acute phase after stroke. And this patient was in San Francisco Bay Area, so he had access to the most, he had maxed out on rehab. There was a lot of rehabilitation that this patient had undergone. And this is the 3D reconstruction, and you can see that there was a supermassive stroke. This entire part of this parietal lobe is gone. But if you see the the DTI, the corticospinal tract, which is shown in yellow, that was still intact. So this is the description of the patient, and the ECOG grid was there. So we made this patient do a very simple task, a center out task, which was visually guided. So there was a computer screen, which was essentially telling him what to do. He had to keep his hand in the center until a cue came. And then one of the 4 directions, a red light came on. And after a variable amount of delay, it turned to green when he had to initiate his movement. Another point to note here is that this is not skill reaching. He just had to make his arm go from center to outside. And these are his trajectories to the 4 different targets. We made him do this with both his affected hand and unaffected arm. And the behavioral parameters definitely indicated deficit in the affected arm. His reaction time was longer when compared to his unaffected arm, and his speed of reaching was also slow with the affected arm. So what was happening on the ECOG grids that we were recording from? So we saw that there was a lot of high frequency activity right around the time of movement onset, which is indicated by time 0 here. The high frequency activity is likely indicative of the spiking activity of individual neurons. So that was there, but this low frequency activity that I was talking about in the rodent, that was very much lacking. This high gamma was high, and this is the mean EMG activity, but the low frequency activity wasn't there. And this is average for the stroke patient in this plot. So then we also wondered that could it be oddity of just one patient? So typically in these EMUs, the other kind of patients are more common. So we also recruited 2 non-stroke patients and made them do the same task. So in the 2 non-stroke patients, we really did not see this lack of LFO. And they were able to, they were more swift at the task, the center out task. So this very much mimics what we saw in the rodent slide that I was showing you earlier, early versus late after stroke. And this is just another way of plotting the same thing, but this is our stroke subject, and this is roughly the central sulcus. And these are the 3 time points, T1, T2, T3, in relation to the movement. T1 is before movement, T2 is at the time of movement, and T3 is post the movement. So the stroke subject really did not have this low frequency power, whereas the non-stroke patients tended to have this power. So this, I think, was a nice concordance of showing that there is something very, very important. This LFO is across species. It seems to be subserving skills, and it seems to be important for your execution of motor skills. So once we had this concordance established, then we wanted to go and see, we have this oscillation now. So can we go and augment it in some way in the stroke recovering rats to somehow take it to a translational stage to see if we can somehow boost this weakened activity. There are different ways to modulate neural excitability. Earlier in the week, you heard of transcranial magnetic stimulation. But what we did here was epidural cortical stimulation. So this is the top view of the rodent brain. We essentially delivered electrical current over the peri-lesional site. So if this is the stroke site, we implanted bone screws, and the ground bone screw was implanted over the olfactory region in the contralateral hemisphere. And we passed a direct current through these sites. This was the ground, and this was either the anode or the cathode. And another thing I want to highlight here is that the electrical field was pretty wide with this kind of montage. The current is essentially going to a lot of places in the brain, not just in the perilesional or near the stroke sites. And another thing to keep in mind is that this is in a smooth brain. So how electric field is interacting with neurons here is going to be very, very different from how if you try the same approach in a gyrated brain. With this kind of montage, the idea of our experiments was that what if we deliver the electrical stimulation, which is indicated by this blue square pulse here, right around the time of movement? And can we augment this, the neural dynamics, so that the limb position, which is weak here, can it be facilitated so that the rats are able to grasp? And what we see this, the effect of this responsive stim is that yes. So in this, we recruited rats that had chronic deficit. So the rats which really were not recovering even a month after a stroke. And what we see is that once we do, so this is an example rat and different sessions in that rat. The sessions that we did with stimulation, all of a sudden his accuracy improved. And this was, this on-demand stimulation was really needed. Uh, this activity-dependent stimulations was needed for even for longer duration. So it did not result in long-term plasticity or long-term changes. So as soon as the stimulation was switched off, the accuracy dropped again. But as soon as, if we tried another session where we provided the stimulation, the accuracy jumped again, and so on and so forth. So in this next batch of experiments, we really tested it in a clinical trial kind of a fashion. So in this task, we essentially make them do this task in this reach box, which is made in-house. So we can control, we have certain parameters. So once the door opens, only then they can reach. So we started the stimulation just half a second before the door opened. So this was sort of a closed loop stimulation. So the stimulation was going on only for a 5 or 7 second period right before the door opened. So we were stimulating the perilesional sites during the time of reaching, and rats were pretty motivated. So they used to reach as soon as the door opened. So the electrical stimulation was on throughout the period of reaching. And then we also did sham stimulation, where there was only a 100 millisecond stimulation pulse, which used to get over before the door opened or the reach could be initiated. And what we saw across 7 rats was the strong effect that with this on the stimulation trial. So this was done in a randomized fashion. Some trials had stimulation, some did not have, some had sham stimulation. So there was a 70% accuracy on the stimulated trials. And all these rats had chronic deficits, so they were not doing good on the task. And we saw this in both the middle cerebral artery occlusion model rats and photothrombotic rats, which are color-coded by these 2 colors here. And then we also wanted to see the effect of timing of stimulation. So in this case, the stimulation was 1 second long, and it was the onset of the stimulation had a certain temporal delay from reaching. And what we saw was that the stimulation onset was really, really critical when it was at least half a second before the movement began. So in this plot, you see that the mean LFP, it starts to, that the quasi low frequency activity starts to engage half a second before the movement starts. And if our stimulation is also tied to that, the accuracy is the highest. And if the stimulation really starts later in the reach, the accuracy, it doesn't really improve the accuracy. So in the red here, you see the success or the accuracy on the task, and in the gray, you see the LFP activity. So the time preciseness of this closed loop stimulation was very, very important. So it really had to be triggered in a closed loop fashion to be essentially augmenting this LFO. So in this next stage of experiments, we really tested this under anesthesia. So this was under ketamine anesthesia, and we tried to see what is really happening to the activity. So this under ketamine anesthesia, there is a predominance of slow oscillations. We tested the activity of single units under this anesthesia, and we saw that when we stimulate the brain under this anesthesia, the low frequency power really goes up, and the spike field coherence of the neurons also goes up. So it's not as much of a change of firing rate of neurons, but the firing activity becomes very, very organized around this low frequency oscillation. So this is sort of a test bed to see the effects, because these experiments are difficult to do in awake behaving animals, like to be doing a stroke, a stimulation, as well as single unit recording. But we looked at the activity in the awake behaving in the LFPs. And in the LFPs, we saw that when the rats had deficit and when the stimulation on trials came on, they had this low frequency power, which was really boosted. So this really shows a pathway which can, so we have a marker which can track recovery, and it's also amenable to get boosted via a relatively less invasive approach of epidural cortical stimulation. So this is the working model that the intact CNS, there is this quasi low frequency activity with skill learning. It seems to organize the neural activity also. And this is related to very smooth, consistent, stereotyped movements. And this goes awry in the injured central nervous system. the LFOs are they become scattered, the spiking activity becomes scattered, and this is also related to the production of inconsistent, non-stereotyped variable movements. But with temporally precise direct current stimulation, we can restore this injured CNS to a state of an intact CNS. And we are really excited about taking this forward and seeing if we can really take this to into a clinical trial and test this in patients as well. So some of the other directions which are being pursued in my lab then are um, to implement this activity-dependent brain electrical stimulation, both awake state, which I already presented to you, but also during sleep state. So I'm not going into the details of that, but there is a lot of sleep-related reactivation of awake activity also. So if you're learning a new sport or a new musical instrument or any kind of new musical skill, sleep has a huge role to play. So human studies have already shown that brief naps can improve your motor performance. And this is also true in a stroke recovering brain. So we are also testing that right now, to what extent the sleep reactivation is playing a role in recovery, and if we can modulate sleep also to have an enhanced recovery. And then another focus area is in one of the beginning slides, I told you that motor actions are essentially instantiated through activity in several regions. So there's striatum, cerebellum, red nucleus also has motor representations which become important after a stroke. So we are also now recording simultaneously from multiple regions. So we are really focused on corticocerebellar networks, which I could not because of the time constraints today, but we see that corticocerebellar areas both develop this low frequency activity with skill learning. And then also we want to see like what is happening in these distributed networks during the course of recovery, and if some other sites can be better for stimulation after a stroke. And then obviously we are also interested in developing circuit or cell specific therapies, so which can be accomplished with optogenetics, stimulation 2.0, if you will. So with electrical stimulation, we don't really know where all is it going, but with optogenetics, we can really do targeted interventions. Uh, for example, in this study that I just showed you, our target was to recruit the perilesional pyramidal neurons in layer 4-5, which are essentially projecting down the corticospinal tracts to spinal motor neurons. But we don't know whether our electrical stimulation was only modulating those neurons or several other neurons, because the electric field was huge. So in the future, we also want to test out cell-specific therapies by modulating only the neurons that we really want to have an effect on. So with that, I'll wrap up. I'd like to thank a few people. I did not really show the new data, but the new data on the corticocerebellar ensembles is being led by people in my lab, Amir, Pearson, Nathan. And we are part of this new Center for Neural Science and Medicine at Cedar Sinai, which is located in the city of Los Angeles. Our lab is housed in this building in the city of Los Angeles. And I really want to thank people at UCSF. So most of the work I presented today was part of this 2018 paper, which was published in Karunesh Ganguly's lab, where I was a postdoc. And these are some of the other authors with whom I've published there. And I would also like to thank the funding sources. Our work is funded by the National Institutes of Health, National Science Foundation, AHA, which is the American Heart and Stroke Association, and also Cedar Sinai's institutional funds. So with that, I can wrap up and take any questions.